Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Single-Cell Multiomics Resolves a Sharp Disease State Shift Between Mild and Moderate COVID-19, and our sponsor is BioLegend. Our panelist today is Dr. Yapeng Su, a research scientist in Leroy Hood's lab at the Institute for Systems Biology. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Su. Please go ahead.
Thanks for the very nice introduction. I'm Yapeng. I'm from Institute for Systems Biology. Today, I'm excited to share with all of you our most recent research using systems biology approach to understand immune response in COVID-19. We all know that our immune system are very important to protect us from all different type of infections we're encountering every day. But there are still so much unknown about how our immune system responds to this particular SARS-CoV-2 virus and how such response correlate with different level of disease severity. So to address this big knowledge gap, here at the Institute for Systems Biology, we decided to take a systems level approach to comprehensively characterize a large cohort of patients spanning across different level of disease severity as quantified by WHO ordinary scale. And so far, we have analyzed around 265 COVID-19 patient samples in comparison with many healthy donors. And for those patient samples, we have collected their blood, analyzed their PBMC, the circulating immune cells, and also the plasma environment. So for the immune cells in the blood, we have performed two different types of multiomic assays. So we first using 10x technology to look at the single-cell multiomics of those immune cells. This will give us, within the same cell, the whole transcriptome, basically the expression of 25,000 genes within each individual cells. And for the same cells, using total 60 everything panel from BioLegend, we're also able to quantify the level of 192 surface marker level within the same single cell. And of course, within the same single cell, we also analyzed the T cell receptor and B cell receptor sequences. And all those three layers of information are all coming from the exact same single cell, and we analyze around thousands of cells per blood draw. And for those immune cells, we also look at their functionality, their functional responsiveness by stimulate them and look at their single cell secretome by looking at their ability to secrete many different cytokines simultaneously. So this single-cell secretome technology is actually originally developed here at the Institute for Systems Biology in the HIS lab and now commercially available at Isoplexis and being well used to characterize T-cell therapy product for cancer immunotherapy field. And recently, many other people have been starting to using it to characterize the functionalities of all different types of immune cells across different diseases. For example, here, we use that to characterize the responsiveness of different immune cell types in COVID-19. And for the same patient sample, we also characterize the global proteomic and metabolomic profiles within the plasma sample for those blood draws. And finally, we integrate those immune cell and plasma cell global characterization within the context of the clinical status of those patients by correlating those with the electronic health record of the same patient. So let's first take a look at the immune cell data. We know that our immune system are very heterogeneous. There is very different immune cell types in our blood, and different cell types are, of course, performing different functions to respond to the virus. And fortunately, using single cell analysis, we can pinpoint the exact phenotype for each of the individual cells we analyzed. As shown here on this U map, each dot represents a single cell, and the color basically represents their cell type annotation. And the cell-to-cell distance on the map represent how dissimilar they are based on the global level of all gene expressions. And you do see that different cell types, the cell with different colors, are actually far apart, and cells with the same color, same cell type, are actually close together, indicating that really different cell types are indeed globally different from each other, and cells within the same cell type are more or less similar. So after annotating the cell type for each individual cell we analyzed, the first thing we did is to look at the relative percentages of each of these major immune cell types across patients with different level of disease severity. As shown here on the box plot in the bottom, you can see that from healthy green to mild yellow to moderate orange to severe red, there seems to be a decrease of T cell percentages, NK cell percentages. Those are all lymph nodes. Those are all lymphocytes. The lymphocyte relative percentages are actually decreasing, and the monocyte percentages are actually increasing when the patients are getting sicker. And this is consistent with the clinical report where people see that the patients are getting sicker, there's a lymphopenia. And we also know that within each of these major immune cell types, there are different subpopulations. They're also heterogeneous within each of them. So in the next following slide, we decided to take a careful look at each individual cell type to see how those subpopulations are changing across different levels of disease severity. And the first cell type we looked at is CD8 T cell, which are the killer T cells. Many of you are probably interested in looking at B cells because B cells are able to secrete antibodies to neutralize the virus. But in fact, CD8 T cells are also very important in protecting us from the virus, because once the virus infected our host cell, got into our host cell, then the antibody are no longer being able to go into goes into our own cell to neutralize the virus. So we need the CD8 T cell, the killer T cell, to kill the virus-infected host cell to further release the virus and for further clearance. So CD8 T cells, very important. And what are the subtypes we're interested in within the CD8 T cells? So we know that when the CD8 killer T cells see the virus, they are able to get activated, basically to change from a naive subtype of CD8 T cell to become an effector, fully activated subtype of CD8 T cell to gain the ability to kill. And we find that in our data, we go from healthy green to mild yellow to moderate orange. There's a decreased percentages of naive subpopulation and increase of fully activated effector CD8 T cells, indicating that when the patients are getting sicker, their T cells, their killer cells, are getting more and more activated to fight the severe virus infection. But interestingly, though, if we go from moderate orange to severe red, instead of keep getting more and more activated, the most severe patients actually start to decrease their level of fully activated killer T cells. And that may partially related to why those patients are so sick. Maybe their T cells, their killer T cells are just compromised to become fully activated to fight the virus. And this also potentially resolves some of the conflicting report in literature where some people find that when the patients are getting sicker, their T cells are getting more and more activated. And some other reports find that in the very sick patient, their CD8 T cells actually getting decreased in the activation status. And those different reports are probably capture different subfractions of severities. And this also emphasizes the importance of why we need to perform this big study to cover the whole spectrum of disease severity, to really have the full picture of this disease. So in addition to this naive subpopulation, effector activated subpopulation in CD8 T cell, there's another subpopulation that many of us are interested in looking at, which is this exhausted subpopulation. What is T cell exhaustion? Basically, after the T cell encountering the virus antigen for a long time, the T cell will eventually become dysfunctional. They lose the ability to kill, and they lose the ability to proliferate, to generate a big army of T cells to fight the virus. So they really become dysfunctional. And actually, this T cell exhaustion dysfunction is actually very important in cancer immunotherapy. And you know that the 2018 Nobel Prize is actually awarded to checkpoint inhibitor therapies, which are aiming to stop the T cell exhaustion from happening, to rejuvenate the T cell, to allow it to have the ability to keep killing the cancer cells. And that actually become very successful in cancer, in killing cancer cells using T cell. So we wonder whether in the context of COVID, do we also see this exhausted subpopulation in COVID patients? And if so, will the checkpoint inhibitor therapies, the Nobel Prize winning work, may be beneficial in treating COVID patients? So we looked at our data as shown here. Each dot is individual T cells, and we are able to find a subcluster of T cell as circled here by the dashed line, this cluster A T cell. They actually have very highly upregulated T cell exhaustion signature. But counterintuitively, this cluster of T cell, instead of lose the ability to proliferate, they actually have the highest ability to proliferate. And this cell also, this subpopulation also didn't fully lose the naive signature. So this is really like a hybrid subpopulation. They simultaneously have exhaustion signature, proliferation signature, and didn't fully lose naive signature. And this subpopulation actually are closely correlated with disease severity. As shown here, the percentage of this cluster cell actually keep increasing when we go from healthy to mild to moderate and to severe. So this is very interesting and disease severity relevant. What makes this subpopulation unique? To address this question, we look at the global transcriptome of this subpopulation in comparison with other T cells. And we find that based on gene set enrichment analysis to look at the pathway functions of the unique upregulated genes in this subpopulation, those genes are actually enriched in the transcription factor MYC or rank 3 downstream target genes. And those transcription factors are actually important in T cell activation. They're actually transiently upregulated in the very early stage of the T cell activation. So this indicating that this cluster 8 hybrid T cell are likely representing a transient cell state during the T cell activation process. And in our data, we also find they are transient in different time points. They are either upregulated only in T1 or upregulated significantly in T2, but not like continuously high in both time points across patients. And when we correlate the percentage of this interesting subpopulation with the clinical data, the electronic health record of the patient, we find that the percentage actually strongly correlated with many of the very interesting clinical status. They correlate with the immature granulocyte positively, and they negatively correlate with the total protein level in the patient and plasma in the patient blood. So we know that immature granulocyte is a sign of very severe infection, so that there's so much severe infection going on, and then the immune system is pumping the immature granulocyte to the blood to fight the virus, and that also positively correlated with this transient early upregulation of this cluster 8 T cells, indicating this is really a sign of very severe infection, which requires the immature granulocyte and also requires the overactivation, the so quick activation of this subpopulation of T cells to fight the severe infections. And in fact, this transient hybrid phenotype is also reported in cancer and other chronic infections, for example, HIV, as Professor John Worry and many other T cell biologists have found, and they probably represent proliferative hierarchy in the continuum of T cell exhaustion. And in fact, the hybrid proliferative exhausted phenotype is not only discovered within the killer CD8 T cell, it's actually also discovered within the helper CD4 T cells. Basically, in the CD4 helper T cell, we also find a subpopulation that have the highest exhaustion signature and also highest proliferation signature and didn't fully lose the naive signature. And this proliferative exhausted hybrid CD4 helper T cell are also positively correlated with disease severity, as shown here on this box plot. We go from healthy to mild to moderate orange to severe red, and they just keep increasing. And you may think that because this proliferative exhausted phenotype is very proliferative. They probably can proliferate and generate a lot of daughter T cells that have the same T cell receptor, so therefore they may be very clonally dominant. But in fact, we find they're not the most clonally expanded T cells. What are the most clonally expanded T cell population? Are actually another interesting population is the cytotoxic CD4 population. So we know that CD4 are generally helper cells, and generally helper cells do not directly kill. But we're able to find a CD4 helper T cell that have the ability to kill. They secrete a lot of the cytotoxic molecules, granzyme B, porphyrin, granulysin, and this is very interesting. And we do know that in rare circumstances, CD4 helper T cells do gain the ability to kill. And that was when the T cells are repeatedly seeing the antigen over and over again. And this is likely indicating that this CD4, this cytotoxic CD4 are likely SARS-CoV-2 specific. They're supposed to recognize the virus antigen. And likely in a very severe patient, the virus antigen is not fully cleared, so that the CD4 helper T cell is just keep seeing the virus antigen over and over again, so that even the CD4 T cell start to gain the ability to kill. And you can see that they are indeed very high in moderate and severe patients, in orange and red. And in fact, more than 95% of the highly expanded T cell receptors are all coming from this novel cytotoxic CD4 subpopulation. So you can see that there are two very different but both interesting subpopulations of T cells we find in the COVID patients. And we wonder what are the relationship between these two different subtypes of CD4 T cells. To study that, we projected each individual CD4 T cell onto a functional map, two-dimensional functional map, where the x-axis represents the Th1 signature score, and then the y-axis represents the cytotoxic score, and each dot represents each individual cell. And you can see there are four plots. In the bottom left plot, you can see that the color actually represents what are the subpopulation are these T cells from. For example, red meaning cluster 5 T cell, which are the cytotoxic T cell, the cytotoxic CD4 T cell. And the blue, the light blue, is cluster 8, which are the hybrid proliferative exhausted CD4 T cells. And the dark blue, cluster 1, at the bottom left corner, they're actually the naive subpopulation. And the other three plots, we're plotting the naive score, proliferation signature score, and the exhaustion signature score for each individual CD4 T cell. And you can see that the naive CD4 T cell are residing at the bottom of this kind of branching structure. So indicating those are likely the starting place for those T cells. You know, all the CD4 T cells are basically differentiated from the naive starting point, which makes sense. And you also see that this cluster 5 cytotoxic T cell and cluster 8 proliferative exhausted CD4 T cell are actually residing at the two extreme ends of this two branch, indicating that those two novel CD4 T cells are likely the two distinct destinations for a given CD4 T cell to eventually become. And all the other CD4 T cells are either intermediate stage along the top branch or the intermediate stage along the bottom branch. So this really tells us that likely these two very novel phenotypes are two extreme different destinations for a given naive CD4 T cell in the context of COVID. And interestingly, if we look at the T cell receptor sequences from those two subpopulations and look at how they share with other subpopulations, as shown here on the right-hand side, you do see that these two phenotypes, class 5 and class 8, as shown on the rows, they actually have very different sharing patterns, indicating that their T cell receptor sequences are likely also different. And this may further explaining why those different CD4 T cells are eventually become these two different destinations. Maybe that's partially related to how their T cell receptors are interacting with their related antigens. So we have looked at CD4 T cells and CD8 T cells. Those are all the adaptive immune cells, the one that will be able to remember the T cell, remember the viruses, and later protect us when we see the same virus again. But what about the innate immune cells, the one that generally do not remember? So one of the innate immune cell we looked at is monocyte. Because many people are suspecting monocyte are the troublemaker, which partially contribute to the cytokine storm that have been reported in many of the very sick COVID patients. Because they are suspecting the monocyte and the related macrophages are able to secrete many of the inflammatory cytokines, and which then contribute to this hyperinflammatory symptoms in the severe patient, which are what people call the cytokine storm. So we wonder whether the most severe patients, do their monocyte secrete a lot more of the inflammatory cytokines. And when we looked at our data, we actually find the opposite. The most sick patient, their monocyte actually secreting the least amount of pro-inflammatory cytokine, for example, TNF-alpha, and their monocyte also lose the ability to present antigens to T cells, to educate the T cells. So the very sick patient, their monocyte seems to be dysfunctional. They lose the ability to secrete pro-inflammatory cytokines. They also lose the ability to present antigens for T cell education. And that's very similar to what sepsis field called the immunoparalysis. So, sepsis is a completely different type of infections, and we find in COVID, we also find this type of immunoparalysis phenomenon. And this dysfunctional monocyte percentages are actually significantly increased when we go from healthy to mild to moderate orange to severe red. And in fact, this dysfunctional monocyte percentages is actually closely correlated with the cytokine storm level in the patient, as shown here on the scatter plot. The x-axis basically represent how the monocyte lose ability to present antigens, and then the y-axis represents the IL-6 level in the plasma in the same patient. Basically, IL-6 is the gold standard marker for cytokine storm, and the the drugs that treat cytokine storm is actually targeting the IL-6 and this downstream signaling. And there seems to be a strong correlation there. So this indicating that there must be a close cross-communication between this monocyte behaviors and the plasma profiles. And the people rationalize it, likely because we already have so much pro-inflammatory cytokines in the very sick patient, so that we don't want the monocyte in the same patient to secrete more pro-inflammatory cytokines to make the patient worse. And that's why this monocyte lose ability to secrete those pro-inflammatory cytokines. But in the same time, they also lose ability to present antigens to educate the T cell, which may be a bad thing, and which also may explain why in the very sick patient compared with moderate sick patient, their T cells are decreased in activation status. So this close communication between the plasma and the monocyte behavior triggered our interest to systematically analyze the global plasma-omic profiles and its connections with the global transcriptome profiles of the immune cells monocyte. The pathways that enriched for the global proteomic profiles that positively correlated with severity are actually independently matched to the pathways that enriched for the transcript that significantly correlated with disease severity in the patient. So then the enrichment of pathways of these two very different data sets independently match to the same functional pathways, indicating that there is a close communications and coordination between the global plasma proteomic profiles and the global monocyte transcriptome profiles. So those are the monocyte analysis. We also find a lot of interesting analysis result from B cells and from NK cells. For example, we find when the patients are getting sicker, their B cells have more antibody secreting plasma cells. And for the NK cell, which also have the ability to kill the virus infected host cell, their proliferation ability actually getting more and more increased when the patient are getting sicker. Likely, in the very sick patient, we require the NK cell to generating a big army of NK cells to fight the virus. But because of the time limitation, I cannot cover too much of the details of the B cell and NK cell analysis. What I do want to focus on for the rest of my talk, though, is the completely different angle to look at our immune response to COVID-19, to SARS-CoV-2 virus. So in all of my previous slides, we have been talking about all different type of immune cells in separately. But if you think about it, our immune response to this virus are supposed to be a symphony, where different cell type are a different type of instrument. They play different type of music, but they all have to play together in harmony to have the best music. And similarly, our different immune cell types are performing very different functions, but they all have to coordinate in order to best respond to this virus. But the question is, how? How do these different cell types coordinate with each other in the context of COVID? And in other words, how the expression of thousands of genes within all those different cell types co-regulated across different patients with different levels of disease severity. This question is not easy to address simply because the enormous amount of data we're studying. As you can think about it, we have around 200 to 300 samples. And for each of the patient sample, we have many different types of immune cells. And within each of this immune cell type, we have expression of thousands of genes. That's millions of data points. How do we simplify that? Well, that's the beauty of systems biology. Systems biology are good at simplifying the complexity, and it can potentially simplify a very high-dimensional data into just one dimension, which can essentially project the comprehensive picture of each individual patient along that axis. And in fact, we are able to achieve so using systems biology approach to project the patient immune cell data set to a single variable, which reflect how different cell types co-regulated across all different patients. And all this analysis are performed purely based on the immune cell behaviors. We didn't consider any of the clinical data yet, but later when we check back how different clinical data of those patients project back onto those single coordinated immune response variable. And we do see that when we go from healthy green to moderate yellow to healthy gray to mild yellow to moderate orange to red severe, this seems to be gradually moving from left to right, indicating that there is close connections between the coordinated immune response and the disease severity. And there seems to be a big jump between the mild yellow to moderate orange, indicating that there must be a very strong, sharp immune response reconfigurations from mild patients which do not require hospitalization to moderate patients which start to require hospitalization. So in addition to disease severity, we also look at many other clinical data from the electronic health record and how those data correlated with this coordinated immune response module, as shown here on this plot, module 2, and their correlation with all those columns, which are all different type of clinical metrics from the patient clinical health record. And you find that the red color represents the positive correlation, and module 2 correlate with C-reactive protein, which are a sign of inflammations. So when we go along this coordinated immune response modules, there is an upregulation of inflammation. But also interestingly, many of these coagulation factors are actually also strongly correlated with this coordinated immune response module, implying that many of the patients are suffering from those clotting problems are likely have some immune connections. And at the bottom, we also look at how the global plasma proteomic and the metabolomic profiles correlated along this coordinated immune response module. So the immune response module didn't compute it from the plasma data, but when we later check the correlation, we find there is a strong connection there too. When we go along this coordinated immune response module, there seems to be increase of inflammation markers and immune activation markers in the plasma accompanied with the tissue damage markers. And in the plasma, we also find a significant loss of many nutrients. For example, many of the lipid and essential amino acid species are actually decreased when we go from left to right along this coordinated immune response module, and indicating that there's a communication between the plasma environment and the immune cell behaviors across patients. And finally, we can also see that how those different immune cell types are changing along this coordinated immune response module, as shown here on the map, on the heat map. There's the CD8, CD4 T cell, B cell, and monocyte, and NK, and how their transcriptome, surface marker level, and pathway scores are changing from left to right along this coordinated immune response modules. And basically, we can see that when we go from left to right, the T cells are getting activated. The B cells are also differentiated into the plasma antibody secreting cells, and monocyte is getting dysfunctional, and NK cell is getting more and more proliferative. And the many signaling pathways and metabolic processes within all those different cell types are also changing along that coordinated immune response module. And importantly, many of those novel phenotypes we discussed earlier are actually strongly correlated with this coordinated immune response module. Those proliferative, exhausted T cell phenotypes, those cytotoxic helper CD4 T cells, dysfunctional monophenotypes, they are all significantly positive correlated with this coordinated immune response module and start to appearing from mild to moderate disease state transitions, indicating those immune dysfunction are actually closely co-regulated from patient to patient, and there is definitely a sharp disease state shift and immunological reconfiguration from mild to moderate COVID-19. And this analysis tells us that really our immune response are a symphony where all different cell types coordinating with each other to respond to the SARS-CoV-2 virus. And only by using systems biology approach can we really simplify such complexity to this clean and unify the single coordinated immune response module to really understand how they coordinate with each other. And such immune coordination is actually strongly correlated with disease severity and showing a sharp disease state shift from mild to moderate disease. And because of the time limitation today, that's so much I can cover, but there's also a lot of other interesting findings we have that we don't have time to cover today. So I would encourage the audience to read our paper on cell. And finally, I'd like to thank my faculty mentors, Professor Laurie Hood and Professor Jim Heath, for the mentorship on this exciting systems immunology project. I also want to thank my close colleagues from Institute for Systems Biology and Swedish Medical Center for working closely and intensively with me together on this project days and nights. Without all of their effort, we really cannot achieve such big study within such short amount of time. I was also so fortunate to work closely with essentially the world's leading immunologists to understand the COVID immune response, including Professor Mark Davis from Stanford and Professor Phil Greenberg and Rafiko Tartar from the Fred Hutch Cancer Center, Professor Jeff Bluestone and Professor Louis Linney from UCSF, and Professor Alan Adam from Seattle Children's Hospital. I also want to thank our colleagues and collaborators from UW for the support on the sequencing and, of course, the funding. Without any of the funding source, we really cannot perform this study. And with that, I'd like to thank all of you for your attention, and I'm happy to take any questions. Thank you.
Thank you, Dr. Su. As a reminder to webinar participants, if you do have a question, please type it into the Q&A box in the control panel. We'll begin the Q&A. All right. So, Dr. Su, how do you classify naive and effector CD8 T cells? Did you classify those based on clustering, or was that site-speak data? And how did you consider the memory CD8 T cell group?
That's a great question. We actually start with the whole transcriptome of our 10x data, which give us, a global transcriptomic view of the T cells. And we use those to cluster the T cells into different subtypes globally. And then later we use a lot of those known markers to annotate what are the meaning of those different transcriptome unique clusters. And regarding the score, we actually use a group of well annotated gene set. Like for naive, we have many naive cell gene signatures. We can just consider them all. And for exhaustion, similarly, we use like PD1, TIM3, LAX3, and all of those canonical exhaustion markers to define those scores. And I hope this can clarify some of the detailed analysis. But we also provided all the detailed analysis in our master section of the paper. So if the audience are curious about the nitty-gritty details, it should all be there.
Great. And did you follow the patient cohort prospectively, or were there single time point measurements of patients in their disease? And if you did follow them prospectively, did you see any markers that indicated whether the patients were at risk of developing severe COVID?
That's actually another great question. In this study, we basically collected 2 different time points per patient. And the first time point is very close to the clinical diagnosis of the patient. And the second point is 7 days later. And in this particular study, we haven't looked into too much about how the early markers may be correlated with the later disease severity change over time. But that's definitely something we can, or any of the few people can further do using our data to analyze any of the predictive marker early on that can indicate the progression of the disease severity.
In terms of the monocytes, did you notice any changes in the composition of monocyte subsets according to CD14 or CD16, such as classical, intermediate, or non-classical? For example, was one subset more immunoparalyzed than another?
That's a great question. Yes. By looking at the transcriptome of the monocyte, the most obvious difference we see first is the separation of classical and non-classical monocytes, which are having different CD16 and 14 levels. And we do find that when the disease, when the patient are getting sicker, the non-classical monocytes are decreased. And you know that the non-classical monocytes are expressing higher HLA class 2 genes. So those are also matter as antigen presentations. So those are also potentially contribute to the impairment of T cell adaptations. in the severe patient. And the dysfunctional immunoparalyzed monocyte subpopulation are actually one subpopulation within the classical monocyte.
Could you comment on how a cytokine storm response could be controlled in individuals with a previous memory to COVID-19 exposure or infection?
That's actually an interesting question. I think let's first talk about how do people generally control cytokine storm. Because cytokine storm is one of the very severe symptoms that a lot of severe patients are suffering. And in the CAR T cell field, actually, many of the cancer patients who receive the CAR T therapy, they also suffer from cytokine storm. And what people have been using to control cytokine storm in CAR T is to use IL-6 inhibitors to inhibit IL-6 signaling, which turns out to be very useful in the CAR T field. And there are recent clinical trials using the IL-6 signaling inhibitors to treat COVID patients, and it seems to be very successful. So those are generally how we control the cytokine storm in COVID patients in general. But then the question is, do we need to control cytokine storm for the patient who have this immune memory to SARS-CoV-2? For example, the vaccinated people, which they are supposed to get the immune memory response to the virus. And likely we don't really even need to, because according to the SARS-CoV, many of the study on the vaccine people follow up, they find that although the vaccine may not necessarily prevent the infection totally, in the future, it at least can significantly control severe infections, meaning that even if the patient the vaccinated person are getting the virus infection, it will be less likely to develop a severe COVID-19. So maybe it's not necessarily possible, not necessarily needed to control the cytokine storm, because it's just less likelihood for the vaccinated individuals to develop such a severe COVID-19 symptoms like cytokine storm.
Have you noticed that behaviors of CD8, CD4 effector T cells increase the cytokine storm? And were there any differences in the levels of these cells in severities of COVID in different patients who had different severities of COVID?
So that's an interesting question. Like whether the cytokine storm are contributed by those cytotoxic T cells, or they are contributed mostly from macrophages, monocytes, those myeloid cells. Those I don't know the exact answer, but my guess is more likely the myeloid cells. But then that doesn't guarantee the cytotoxic T cells are not necessarily contributing to the pathology in the patient. Some people are suspecting the activation of cytotoxic helper CD4 T cell may be detrimental, but some other people are saying it could be beneficial. The reason why some people are suspecting it could be detrimental is because the CD4 are interacting with antigen-presenting cells. And once the CD4 helper T cells gain the ability to kill, they are possibly able to kill the antigen-presenting cells that they're interacting with. And the killing of antigen-presenting cells may be bad, because then that should be detrimental to have the antigen-presenting cell to present antigen to educate the T cells, to educate an adaptive immune system. But there are also other people saying that those cytotoxic CD4 may be beneficial too. So there are still definitely a lot unknown questions in the immune response in SARS-CoV-2, and many of the immunologists are actively investigating those questions.
Could you comment on the type of T-cell response elicited by vaccines that you think could be the best predictor of protection from disease after exposure to the virus?
Sorry, can you repeat the question? I didn't catch the middle part.
Yes. What type of T-cell response elicited by vaccines do you think could be the best predictor of protection from disease after someone is exposed to the virus?
I see. So we haven't studied the vaccination populations. All we studied here is, the natural virus-induced immune response. And there are definitely a lot of differences regarding a natural virus infection-induced immune response and immune memory versus a vaccination reagent-induced immune response, for example, T-cell response or B-cell responses. And because we didn't investigate the vaccination population, so it's hard for me to comment on the vaccine immune response and what could be a good predictors. But that will be definitely a great question that the field should investigate into. Was your study done for any specific COVID variant? And do you think that your conclusions might change with any of the new variants that have been discovered? That's also another great question because, there's different variants of the virus, which raise a lot of concern in the field that if we develop the immune memory to the previous virus version, then if there's a future variant, can our immune response be able to still control the future variants of the virus? That's actually another question that the field have been intensively investigating. In our current study, we didn't sequence the viral genome from these hundreds of patients. So it's hard for us to know which of the virus genome are in each individual patient and whether those correlated with the virus infection. But in some other study that we are participated in, we do find that, in the reinfection case. In one of the reinfection case, there is the patient whose second infection is actually a different virus strain than the previous one. And we do find that the second infection is definitely much milder than the first infection. That may imply maybe from the first infection can somehow at least control the severity level of the second infection. But there are definitely a lot more unknowns which the field should be investigating into how different virus variants and how different immune memory are in controlling different virus variants.
Great. And could you tell us if your data is currently being used to monitor patients or triage patients? And as a follow-up to that, could your data be used to identify any potential points of intervention or to identify any currently approved drugs that could be used for treatment?
Do you mind repeat your first part of the question?
Sure. The question was if your data is currently being used to monitor patients or to triage patients.
And then the second question is
if you think your data could be used to identify potential points of treatment or intervention.
I see. For monitoring the patient, it's maybe hard to use in real time because all of this very deep analysis require time to perform all of this sequencing experiment. So if we want to real-time monitor the patient status, probably we don't have the time to wait for a month to get the single cell data back. to analyze how the people are doing a month ago, but our data set do provide a deep dive, a deep view of what's happening deeply in all different parts of the immune component. One thing we could possibly use to monitor the patient status is the plasma omic measurement. Those things are generally easier and quicker to quantify. And there are definitely studies trying to use the plasma protein or metabolite as a metric to evaluate or to predict the patient's survival later. So that could be something people can study using our data to see whether they can predict the future disease progression, and then maybe validate that into a different cohort to see the same set of marker would be able to predict and monitor patient disease future progression later in a different cohort. And regarding the treatment, definitely there are a lot of insights that can potentially be drawn in the big data set we have collected. And one of the perspectives that some people are looking at is the metabolic differences in the patient. So basically, we see that in a very severe patient, there are actually a decrease of many of the nutrients, the lipid and the essential amino acid are actually decreased in the very severe patient in their plasma. And people are suspecting whether there's certain nutrition you should provide to the severe patient to further boost the immune response to fight the virus. But there are definitely a lot we need to dive into to fully find the best treatment or even the nutrient supply that we should give to the patient to help the patient better fight the virus.
All right, thank you very much, Dr. Su. We've reached the top of the hour, so we will wrap it up there. We would like to thank Yapeng Su and our sponsor, BioLegend. If we didn't have time to get to your question, we will try to follow up with our expert. As a reminder, we have a pop-up survey that will populate after you log out, so we ask our attendees to take that for us. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this genome webinar.